Hi, welcome to today's AP Biology lesson. My name is Allison Kate, and I'm teaching today from my home in El Cerrito, California. And I thought that I would give you a little geography lesson before the biology lesson today. For those of you who may not be sure where El Cerrito is, it is about 15 miles east of San Francisco and about 5 miles north of Berkeley. So from my table today, I can look out and see the Golden Gate Bridge, and it's a beautiful day here. So thanks for being with us. I usually start, for those of you who have been tuning in, by talking about the 2020 exam details. And today I thought I would skip that and just jump right into our lesson. For those of you who are just joining in for the first time today, you can see our recorded lessons, and all of that information is in those lessons. So let's get started with our exam overview. Okay, excuse me, our lesson overview. So today, maybe. Awesome. All right, there we go. Today we are going to be reviewing topic 2.8, which is on tonicity and osmoregulation. And these are traditionally very difficult topics. So I'm going to give you lots and lots of practice. We're going to work on science practice 4, which is constructing a graph, and science practice 5, which is performing mathematical calculations. And we're going to be working today with 2 formulas. These are from the AP Biology formula and exam sheet, and they have to do with water potential. So don't forget your pen and paper. And also today, you would probably be happiest if you had a calculator with you. Before we embark on today's lesson, you should be familiar with the principles of diffusion and osmosis and cell membrane structure. And you should also be familiar with the terms hypertonic, isotonic, and hypotonic. And I'm going to give you lots of practice with those today as well. By the end of this lesson, you will be able to explain how concentration gradients affect the movement of molecules across membranes, explain how osmoregulatory mechanisms contribute to the health and survival of organisms, and construct a graph. So we're going to get started with our warm-up, and you know that this is a question that was given from the previous lesson. So my colleague, Ms. Pfizer, gave you a lesson yesterday on membranes and facilitated diffusion, and she asked you for homework to discuss the organization of the genetic material in prokaryotes and eukaryotes. And she gave you a hint. She told you to think about what you know about the nucleus and think outside that area. Okay, so if you didn't do this last night, be thinking about it. Maybe you can jot down some ideas. And if you did do it, grab your homework so you can compare your answer to the scoring guidelines. So first, let me explain how this was scored. This is worth 4 points. You got 1 point for each bullet, but you had to have each pair, right? You had to have a bullet from a prokaryote and a eukaryote in order to earn that point. So I think you'll see what I mean when I go over this first one. So if you talked about no introns being present in prokaryotes, you also had to say that introns are present in eukaryotes. In prokaryotes, as we all know, the DNA is not in the nucleus, but rather in a nucleoid region. In eukaryotes, the DNA is in a nucleus separated from the rest of the cell by a nuclear membrane, but it is also found in both mitochondria and chloroplasts. Prokaryotic DNA is not surrounded by histone proteins, whereas eukaryotes is, so does contain histone proteins. Prokaryotes generally only have 1 chromosome, whereas eukaryotes have more than 1 chromosome. And finally, plasmids, or extracellular pieces of DNA, are common in prokaryotes and they're rare in eukaryotes, although they are found in yeast. All right, so I hope everybody got a 4 out of 4 on that one. So here's what you need to know about today's topic. First, let me tell you what tonicity is. And then we'll talk about how it relates to osmoregulation, or maintaining an osmotic balance within your cells and body tissues. So tonicity is a measure of the effective osmotic pressure gradient. It's the water potential of 2 solutions that are separated by a semi or selectively permeable membrane. Another way to say that is that it's the relative concentration of solutes dissolved in a solution, which determines the extent and direction of diffusion. So if you look at my image, I have a cell membrane, and just to review that cell membrane structure, you can see the phospholipid bilayer. So the phosphate heads are represented by those pink circles, and they are polar, and the fatty acid tails are pointed toward each other, and they are nonpolar. So those phosphate heads point out toward the outside of the cell and in toward the cytoplasm. And those two solutions that the cell is suspended in may have different amounts of dissolved solutes. So that is what determines the tonicity, which will then determine the way that materials move across that cell. In the middle of this cell membrane, we have an integral membrane protein, which is called an aquaporin. And I tell my students that this is just like a little water slide, right? So the polar water molecules, which are small, can easily pass through this pore in this integral membrane protein because it is a polar channel for the polar water. So now let's look at the actual statement from the course and exam description. It tells you that external environments can be hypotonic, hypertonic, or isotonic to the internal environments of cells. And we're going to define those terms and give lots of examples throughout this lesson. Then it tells you that water moves by osmosis from areas of, and then you have some slashes. And I honestly think that this is why this topic is confusing to students, because there's lots of different ways to say the same thing. So we can say that water moves by osmosis from areas of high water potential to low water potential, or we can say it moves from low osmolarity to high osmolarity, or we can say that it moves from low solute concentration to areas of high solute concentration. All of those things are saying the same thing, but they're saying it in a different way. Okay, so when I teach this to my students, I always tell them that the most important thing to remember is to remember where water is, and water will always move from where there is more water to where there's less water. And we'll see examples of this in relation to water potential, osmotic potential, and solute concentration throughout today's lesson. So for a science skill, we're going to practice doing some calculations. The first relevant equation is the equation for water potential. Water potential is represented by the Greek letter psi, kind of looks like a little pitchfork, and it's got two components. Psi sub p is the pressure potential, and psi sub s is the solute potential. And again, we'll be talking more about these when we get into the lab activity that has to do with water potential. So let's take a look at these terms: hypertonic, isotonic, and hypotonic. So hypertonic comes from the Greek hyper, meaning over, and tonos, meaning stretching. Here's the definition. It's any solution that has a higher osmotic pressure than another solution. It refers to a solution that has more solute than another solution. So when I'm teaching this to my students and I have them imagine a solution that's filled with lots and lots of sugar. Sugar is a solute. And if you have lots and lots of sugar, let's say you drank that solution, you'd be super hyper, right? Because there's more sugar than another solution. Okay, so now keep that in mind, because now we're going to talk about a hypotonic solution, which is any solution that has a lower osmotic pressure than another solution. This refers to a solution that has less solute and more water than another solution. So now imagine just drinking a glass of distilled water. It is pure water, so there is no solute. That would be a hypotonic solution. Now notice that I'm always talking about these in relationship to another solution. You can't simply say that a solution is more hypertonic, right? You have to compare it to another solution. Finally, an isotonic solution. Iso means same, so it refers to two solutions that have the same osmotic pressure across that membrane. And when you have that, it allows for the free movement of water across the membrane without changing the concentration of solutes on either side. So there's movement, but you're not changing the solute concentration. My image shows you both an animal and a plant cell in the three different kinds of solutions. So let's look at hypertonic first. So if we placed an animal cell in a hypertonic solution, the hypertonic solution will cause the animal cells to lose water because they have less solute, more water than the hypertonic solution. So the water will leave the animal cell where there's more water for the hypertonic solution where there's less water, and the cells will shrivel. Another term for that is crenate. In a plant cell, notice that that plant cell has a cell wall around it. And if you place a plant cell in a hypertonic solution, it will actually lose water to the environment, and we call that being plasmolyzed. And that's if you think about a plant cell that's growing in the soil, if the soil is too salty, let's say that there's been a drought and there's not enough water, or maybe there's been some agricultural runoff that has a lot of salt in it, then the surrounding area is hypertonic, then that plant is going to lose water and wilt, okay, because its cells become plasmolyzed. An isotonic solution, as mentioned, the concentration of solute or water is the same across that membrane, so you shouldn't see any change in the size or shape of that cell. And you might, if you watch any medical shows or doctor shows, they talk about using an isotonic saline solution if they're trying to give an IV to a patient. And that is telling you that saline is salt and our actual body fluids are a little bit salty. So they do need to be isotonic. So if you're getting an IV and they don't put in an isotonic solution, then your blood cells can either shrivel or swell. So that's really important. In a hypotonic solution, your cell actually has more solute, right? There's dissolved solutes and proteins and such. So if you place your cell into a hypotonic solution, water will flow in and the cells will become swollen. They could even burst or lyse. And if you look at a plant cell, again, that cell wall, when water flows in to a plant cell, it actually makes that cell turgid or stiff, and that's how the cell maintains its turgor pressure so that it doesn't wilt. A loss of turgor pressure is wilting. Okay, so hopefully that gives you a working understanding of these three terms, but we're going to use these throughout this lesson. All right, so first let's read the statement from the CED. It says that osmoregulation maintains water balance and allows organisms to control their internal solute composition or water potential. The formula, and we're going to get lots of practice with this formula. I know it looks scary. It says psi sub s is equal to negative I C R T. And I'm going to go through what those are, but it reminds me that several years ago on an AP Bio FRQ, this question was given and the students were asked to describe what each of those components mean. And you got to love the students. A lot of them didn't know this, and so they entertained us by saying that it means I can't really tell. So none of you are ever going to be in that position because I stands for the ionization constant. That is the solute in the solution, its tendency to ionize. So an example would be if you were looking at a sodium chloride solution. We all know that sodium chloride will ionize in water into sodium ions and chloride ions. If you put a sugar into water, it does not ionize. So that is a value that tells you whether or not the solution will ionize. C is the molar concentration or molarity of the solute. And you'll see when we get to our lab that there is a way to determine the molar concentration of a particular cell in a particular solution. R, you may recall from chemistry days, it is the pressure constant, and you may know it from the ideal gas laws, PV equals nRT. It's the same R, and it doesn't change, and it's listed there. You would never have to remember this, but it's 0.0831. It has weird units, liter bars per mole degrees K, and the T is for temperature, and the temperature is in degrees Kelvin. And so when we do these activities or experiments in our lab, the thermometer that we use will measure in degrees Celsius. So you always have to remember when you're using this formula to convert the degrees Celsius to degrees K by adding 273. All right, so what do these images represent? I'm showing you some onion cells in a hypotonic solution. So onion cells are going to have some dissolved solutes in them. These cytoplasm have been stained with carbofuscin, so they look pink. You pop them in a hypotonic solution, and water is going to move into the cytoplasm and cause them to swell. And then we have a single-celled organism. This is a paramecium, and it lives in a freshwater environment. So you might want to think about the consequences of maintaining osmotic balance for that organism. If it's salty, right, because it's got dissolved solutes and it lives in freshwater, what's going to happen, right? The water's going to move in. So what does that paramecium need to do to prevent itself from blowing up? And I'll give you a little hint. I've pointed out structures called contractile vacuoles. Okay, so let's take a look at this slide that from the CED tells us that growth and homeostasis are maintained by the constant movement of molecules across membranes. So in this case, the homeostasis is of the osmotic balance in your fluids and cells. So just to follow up on the paramecium, remember their cytoplasm is hypertonic to the environment they live in because it has solutes dissolved in it. So that means that water is going to rush in by osmosis. So what happens to prevent that paramecium from taking on water and blowing up is that those contractile vacuoles act as pumps, and they pump out that excess water, kind of like if you were in a canoe that was taking on too much water, you would bail it out. So those contractile vacuoles bail out the excess water and allow that paramecium to maintain homeostasis in terms of its osmotic balance. Let's take a look at both a freshwater fish and a saltwater or marine fish. So freshwater fish obviously are kind of like the paramecium, but they're multicellular. So they have solute hypertonic concentration compared to the water that they live in. And they have two adaptations that help them maintain their osmotic balance, which is they have super efficient kidneys, so that excretes the water quickly, kind of like those contractile vacuoles, but they also reabsorb the salt that's in their urine before they excrete their urine. And then that minimizes the losses and actively takes salt from their environment. By bringing in that salt can make them minimize the loss of that salt in their cells. And then finally, in a saltwater or marine fish, it has an internal osmotic concentration lower than the surrounding seawater, right? So it's hypertonic, sorry. So it tends to lose water and gain salt. So this adaptation has a way to actively excrete salt out from its gills. So in both the freshwater fish and the marine fish, you see that there are special cells in the gills that maintain their osmotic balance. So just want to see if you're paying attention and see if I can get you to stay to the end. This is topic AK. Those are my initials, and I have a multiple choice trivia question for you. I don't know if you guys are on Facebook, probably not. It's more for old people like me, but there's a thing going around that has you list 5 scientists that you've met, only one is not true. So I'm going to give you a multiple choice and you have to choose the correct answer. And if you stay tuned to the end, you'll see if you were right. So your first choice is Dr. Michael Nachman. He's of rock pocket mouse fame. If you've been tuning in for a while, this may be a giveaway. James Watson, the co-discoverer of the structure of DNA. Rosalind Franklin, who took the X-ray crystallography photograph of DNA that led Watson to make his discovery about the structure. Dr. Bonnie Bessler does research in quorum sensing and bacteria. And Dr. Arthur Kornberg discovered DNA polymerase from a thermophilic bacteria. So I have met 4 of these 5. Which one didn't I meet? All right. So let's take a look at labs now. And there are 3 parts to the diffusion and osmosis lab, which is investigation 4 in the AP Biology lab manual. Ms. Pfizer went through the diffusion part with you yesterday. Today I'm going to take you through the osmosis lab, which uses dialysis tubing, dialysis bags, and then we'll also take a look at the water potential lab that uses potato cells. So in the first one, we are going to investigate osmosis using dialysis tubing. And we're going to look at the relationship between solute concentration and the movement of water through this selectively permeable membrane by the process of osmosis. And I know that you've probably done this lab or seen this lab, but I want to just tell you a little bit about the dialysis tubing. So dialysis tubing is used in kidney dialysis, and this is where patients who have kidney problems will go and they will actually have their blood cleaned of any excess toxins and salts through this dialysis tubing. And the way it works is they have microscopic pores in them that prevent the excretion of large particles or the movement of large particles through these pores, but prevent small molecules and water to move across them. So what we do is we take this tubing and we cut it into strips, tie a knot in one end, fill it with our solution, and then tie a knot in the other end, and it creates a bag. And then we can use these as a model membrane system to look and see how materials move across that membrane. So a little bit of review. Remember that we're going to be using isotonic, hypotonic, and hypertonic in this lab. Isotonic means that the solutions have equal concentrations of solutes, and so you're not going to see any net movement in one direction. So there'll be no net change in the amount of water in either solution. If 2 solutions, one has more solute, it's going to be hypertonic to the one with less solute. And if a solution has less solute, it's hypotonic to the one with more solute. Again, these words can only be used to compare solutions. So let's get a little practice with this. And this is a technique that I use with my students. I still do this to this day, even though I've been teaching about this for 3 decades. And that is to draw and figure out from your drawing what solutions are in terms of their tonicity, and then figure out what direction the water will move. So we've got a figure over here. It's a beaker and a bag that is identified as a semi-permeable membrane. And we are asked to label which solution is hypertonic and which is hypotonic, and then use arrows to show the initial net movement of water. Keep in mind, water will always move from where there's more water to where there's less water, or from a hypotonic to a hypertonic solution. Okay, so you figure out which is hyper and which is hypo. So the solution in the beaker is hypertonic to the solution in the dialysis bag. And that's because in the dialysis bag, you have pure water, no solute. In the beaker, you have some solute in the solution. So it's going to be hypertonic to the water in the bag. And this means that there's more water in the bag, so it will move from the bag into the beaker. And if you measured that, you weighed it, the mass of that bag will decrease. And that is the technique that we're going to use in our lab. So again, sucrose is hypertonic. The distilled water in the bag is hypotonic. And because there's more water in the bag, water will leave the bag for the beaker. Okay, so here's the lab. You are going to create your dialysis bags. You're going to create 6 dialysis bags. In the first bag, you're going to put 0 molar sucrose solution, which is just distilled water. And then into separate bags, you're going to put 0.2 molar sucrose, and then 0.4 molar sucrose, 0.6 molar sucrose, 0.8 molar sucrose. And finally, in the last bag, you're going to put 1 molar sucrose. And then you're going to mass them and record their initial mass. And then you're going to place each of the tubes into separate containers of distilled water. You're going to let them sit for 30 minutes, and then you're going to take them out and reweigh them. And you're going to calculate the percent change in mass. So before we look at some data, some results of this experiment, I want you to make a prediction about what will happen with respect to the different molarity solution and mass change in the distilled water dialysis bag. So let's start there. In the distilled water dialysis bag that you're placing into a distilled water beaker, what's going to happen? Okay, now make a prediction about what's going to happen in each of the bags that do contain sucrose, starting with the 0.2 and working your way up to the 0.1. Okay. So because the dialysis bags with sucrose solutions are hypertonic to the distilled water in the beaker, water is going to move across the dialysis bag membrane out of the beaker and into the bag. And that means that the bag will increase in mass. The bag placed in the 0 molar sucrose solution will show no change because those solutions are isotonic. All of the bags containing sucrose will increase in mass, but the one that will increase the most is the one with the highest concentration of sugar, because it would be the one that had the largest concentration gradient. Okay, so I want you to think about that, because that prediction is what we're going to look at in our data. So I'm showing you some data from my class, and I always project the data to my students once they have entered it in. And in this particular lab, you can really spot trends and anomalies. So if you look at group 1, beautiful job, group 1. In fact, we know that there should be no change in mass when you take distilled water and put it into distilled water, and they got that. They also have the trend that as you increase in sucrose concentration, you increase in the percent change in mass. So group 1 did a great job. Take a look at group 2. They're off to a good start with their 0 molar. And look at 0.2 molar. Yikes. So there's some type of experimental error, because they have a huge increase in percent change in mass, way over even the 1 molar sucrose solution. So that is an anomaly. Both group 3 and 4 look pretty good. They follow the trend, but they have a little bit of an increase in their 0 molar. Group 5 is great. Group 6, they have the trend, but they have a large change in the isotonic solution. So I'm going to leave this up here. You guys can look at this later. The next step in this lab would be to calculate the means for each concentration, and also calculate plus or minus 2 standard error of the mean. Okay, so let's say we did that and we graphed it. And here's our graph. I want to point out that on the x-axis, you've got sucrose molarity, which is the units or moles per liter. And on the y-axis, we have the percent change in mass. And this is the mean or average of the class. And oftentimes I'll have my students do their group as well as the average, just to see the difference. And I have them graph their plus or minus 2 standard error of the mean error bars. So I'm going to go ahead and just draw in this best fit line, and you can see that the data shows the overall trend, right? That as you increase in the concentration of sucrose in the bag, you increase the mass of the bag. So they got good results. However, if you're talking about statistical significance, if you look at the error bars, right, we do see that there's error bar overlap here. So we know that 2 of the groups, or 3 of the groups, were not right at the isotonic point of 0 here. And then we know that there's some overlap on the point. So not great data, but this is actual student-generated results, and we see the trend. So our hypothesis would be confirmed by this data. So moving on to the water potential lab. The water potential lab uses potato cores, but you can use any vegetable. I know some teachers use carrots, or you could use zucchini, or some teachers will even have you look at the difference between different kinds of potatoes. And I'll tell you that the whole point of this exercise is to determine the molar concentration of sucrose in a potato cell. In other words, you're trying to find out how sweet a potato is. So I often like to have my students do a regular like brown potato and a sweet potato and really ask the question, is a sweet potato really sweeter than a regular potato? And you can figure that out by determining the concentration of sugar or sucrose within these potatoes. All right, so before we get rolling, let's just review water potential. Water potential is that term that botanists use when we want to predict the movement into or out of plant cells. Again, this is super important for plant cells because they can't move to find water. So if you are a plant cell, let's say you're a potato plant and your roots are in that soil, you need those cells to be hypertonic or have a low water potential to encourage the water to move in from the soil through the process of osmosis. So this is a really important concept for those plants. All right, so remember that we're looking at water potential being represented by that Greek letter psi. It looks like a pitchfork, and it has two components as mentioned previously. It has a pressure potential and a solute potential. But I want to point out something to you in this lab. Of course, we know that water will always move from where there's more water, a higher water potential, to where there's less water, a lower water potential. So water potential measures the tendency of water to leave one place for another. One way you can do this is picture that water diffusing down a water potential gradient. By convention, the water potential of pure water at atmospheric pressure is defined as being zero. So the pressure potential is zero. Okay, this means that for this experiment, the water potential is equal to the solute potential. So if you look at that equation and at the pressure potential is zero, then water potential is going to be equal to the solute potential. Okay, one of the things that I know is confusing for students after teaching this for a really long time is that solute potential is always negative, right? Since pure water has a water potential of zero, any solutes will make that water potential lower. It'll be more negative. Okay, so keep that in mind as we do our calculations. Okay, so again, I'm using some data from my class. And so what happens is you put your potato cells, your carrot cells, or your zucchini cells into beakers with the different sucrose solutions, and you let them sit overnight. You do an initial mass and a final mass, and you calculate the percent change in mass. Okay, and in this case, you're going to see some of the potato cells are going to increase in mass and some are going to decrease in mass. You can also, if you want more practice, calculate the plus or minus 2 standard errors of the mean. Remember that it's super important that you take the temperature of the liquid in the beaker because you need that temperature data for your calculation of solute potential. So we can see that the overall mean temperature here is 21.8 degrees C. All right, so let's say we graph that, and let me just point out, we still have the same labels. On the x-axis, it's the sucrose concentration in moles per liter, and the y-axis is the percent change in mass over that 24-hour period. This graph is a little bit different. So we're not really using this to display our data. We are using this to determine the isotonic point, or the point at which our best fit line crosses the zero axis. Okay, and so that would be right there. Okay, because the isotonic point represents the point at which the sucrose concentration inside the potato core equals the sucrose concentration in the liquid. Pretty ingenious, right? So when we find that isotonic point, we just drop down to the x-axis and we can figure out approximately what that molar concentration of sucrose in that potato core is. And in this case, it's about 0.22 moles per liter. Okay. So now we have to calculate it. And I know that looks scary. That's the scary one with the solute potential. So again, I is the ionization constant. We did this experiment in sucrose. So the ionization constant is just 1 because sucrose does not ionize in water. We could repeat this experiment in salt water and we would find that the ionization constant would be 2 because there are 2 ions, sodium and chloride ions. The molar concentration is C, which we just determined from the graph of the potato core data, and we determined it to be 0.22 moles per liter. R is our old friend, the pressure constant, 0.831 liter bars per moles degree K. And our temperature, the only thing we have to remember there is we need to convert our degrees C to degrees K, and we can see we do that by adding 273. And now all we do is plug and chug, right? So all the good thing in that crazy units on the pressure constant, everything is going to cancel, and we are going to be left with units that are bars, which are pressure units. So you can check my math, but you would just go ahead and plug in all those values into your calculator, and you're going to come up with a solute potential, which is always, always, always negative, right, of 5.39 bars. Okay, so let's do some practice. So my first practice is just to see if you're comfortable with the concept of isotonic, right? So this question is asking you the beakers of which dialysis tube are initially isotonic to the distilled water in the beaker. So I want you to look at the initial mass of each of the dialysis tubes, and there are 6. And then you look at the mass after 90 minutes, which would be the final mass, right? So isotonic means they're the same. So we should not see any change in mass, right? So look over your data, look over your choices, and see if you can spot any that do not change in mass. Got it? Correct answer is E, right? The distilled water did not change. It was 11.2 all the way across. Okay, here's some more practice. This is a prediction. So let's say you've got an animal cell and you're going to place it into a 0 molar sucrose solution. That's pure distilled water. So an animal cell has solutes, right? So putting it into a 0 molar solution means it is a, fill in the blank, right? Hypotonic solution. Okay. If you take that same animal cell and you put it into a 1 molar sucrose, there's a lot of sugar in a 1 molar sucrose. That solution would be, yes, hypertonic, right? So what's going to happen? So you place that blood cell into a hypotonic solution, it is going to gain water. Okay, because the blood cell has solute, it has less water. So the blood cell water is going to move in, and the opposite is true in the hypertonic solution. This was a prior exam FRQ, and so you can see the scoring guidelines here. So in the 0 molar, we would predict that the cell will gain water or mass, it will swell, burst, or lyse. And that tells you right there the importance of understanding this concept. You don't ever want to have your red blood cells either blow up or shrivel up. So the reason for this prediction is that the cell is hypertonic to the sucrose solution. It's actually a 0 molar, so it's pure water, and water will move in. For the hypertonic solution, the cell will lose water or mass, it will shrivel or crenate, and that's because the cell is hypotonic to the sucrose solution and water potential is greater inside the cell, so water will move to create equilibrium. All right. One more practice with this, and I know you guys have probably done this with microscope practice, where you take a red onion and you peel off that thin tissue, the epidermal tissue, and you go ahead and put that on a microscope slide. You can suspend it in a little bit of sodium chloride solution, and you can see the original slide in that 1% looks like a brick wall, right? So the cytoplasm is in gray, and you can see the cell walls that are shown in white. Now the student added 15% sodium chloride solution and observe the changes. So after adding it, look at that cytoplasm. It's all kind of shriveled up and pulled away from the cell wall. There's a special name for that I told you about earlier. So hopefully you remember that. So which of the following most directly explains the changes in the cells? Is it because the degradation of DNA in the nucleus of the cells? Is it because of the lysis of chloroplast in the cells? Is it because the movement of water from the central vacuole of the cells into the solution? Or because the movement of sodium chloride from the solution into the cytoplasm of the cells? Okay, so get your answer. And the correct answer is C, right? The water is moving from where there is more water, from where there is less water to, let me say that again. The water is moving from where there's more water to less water, which in this case, in the 15% sodium chloride solution, that's hypertonic. So the water inside the cytoplasm leaves for that hypertonic solution, trying to balance it out, and the cytoplasm shrivels up. And that's called plasmolysis. Okay, so long FRQ practice, super fun though. And maybe you have done this experiment on your own. I usually do this as a demo for my students. If you take a chicken egg, which is the largest single egg cell you're ever going to find, well, I guess you could get an ostrich egg and it would be the largest, but they're a little bit harder to come by. So a student conducted an experiment to determine the molar concentration of solutes in chicken eggs. The eggs were first soaked in vinegar to remove the shells. So the shells are just calcium chloride. The acetic acid in the vinegar just dissolves them. So what you're left with is an intact cell with an intact cell membrane. And now you can use it to watch how water moves across it. So these eggs were then placed in sucrose solutions of different concentrations, and the data are shown in this data table. So you're asked to do 3 different things. The first thing is to select the most appropriate data and construct a labeled graph on the axes provided, and I'll show you what that looks like, to best illustrate water movement into and out of the egg. After you've constructed the labeled graph, explain why there is a difference in the percent change of mass in the eggs in the different sucrose solutions. In part B, you're asked to determine the molar concentration of that chicken egg, okay, using the data from the graph like I showed you, and then justify your answer. And then finally, this brings it around to why this water potential concept is so important in living things. Movement of water and nutrients are critical for the survival of plants. Using the concept of water potential, explain the role of positive and negative pressure in the transport of water and minerals. Okay, so this is what your graph grid would look like if you were taking this exam. So notice that you have a heavy dark 0 axis right across the middle, and that you're going to need to plot the points that show a increase in mass as well as a decrease in mass, and then you're going to use that graph to determine the molar concentration of solutes in the chicken egg. So for the graph, you should have a title. Your axes should be labeled. The X must be molar concentration, and Y may be change in mass or percent change in mass. You want to make sure that you have your appropriate data selected, that the axis and data are correctly scaled, and that the line graph correctly plots those points according to the data that you selected. You draw a best fit line, and in part B, we're going to determine that concentration. So in part C. Part B asks you why the egg would gain mass, and it would gain mass if the solute concentration is lower outside of the egg. In other words, there's more water outside of the egg. Water will move into the egg, causing it to increase in mass. And the opposite is true if the egg is in a hypertonic solution, then water will move out of the egg. So it would decrease in mass. And another way to say this is that water would move from high solute to low solute, or you could say high water concentration to low water concentration. All right, in part C, water moves from areas of high water potential to areas of low water potential, and water potential has those 2 components that are described in the formula of water potential. Now here's the important part. It has to do with transpiration, which is the loss of water from leaves to the atmosphere due to the lower water potential of the air. So there's more water in the plant, there's less water in the air. Water's going to want to leave the plant for the air. And then thinking about properties of water, which is a lesson we had previously, water is cohesive, right? It wants to stick together. So when that first water molecule floats off, evaporates off the surface of the leaf, then because of cohesion, it's going to pull that chain of water behind it. The pressure in the xylem is negative, okay? And finally, something called guttation has to do with root pressure, which is a positive pressure. It gives it a little push to get it into the xylem, and from there, that cohesion of water molecules and the adhesion of the water molecules to the xylem tissue is what brings that water all the way up to the surface of the leaf and out through the process of transpiration. Okay. So, quick check for understanding. This is something you can do on your own. I've given you some data. Again, it's my class data, and it's asking you, using the mean class data, construct a graph to determine the molar concentration of the potato cores at 19.6 degrees C, and then determine the solute concentration of those potato cores. So you would graph your data, draw your best fit line, right? Making sure that you've got all your axes labeled correctly. And then find the isotonic point, the point at which the line crosses the x-axis. You can see it's super close to that 0.4 molar, and that would allow you to determine the concentration of sucrose. Oh, let me go back, because then what you would need to do is plug in your values, and don't forget your temperature value, and then determine the water potential, which is just equal to the solute potential. Okay, and the answer is there down there at the bottom for you to see after you try this on your own. Okay, so our task verb today was construct. By now you should be able to construct a graph, and this image is just showing a bunch of different kinds of graphs. Today we practiced just a line graph. We did also look at some error bars to represent the error of the mean. But we also used a graph that we were able to use not just to display our data, but use it to determine that molar concentration of sucrose. All right, so our debrief. You should now be able to explain how concentration gradients affect the movement of molecules across membranes. You should be able to explain how osmoregulatory mechanisms contribute to the health and survival of organisms, and you should be able to construct a graph. Okay, your key takeaways. Cells have membranes that allow them to establish and maintain internal environments that are different from their external environment. Also, osmoregulation maintains water balance and allows organisms to control their internal solute composition and water potential. Some common errors and misunderstandings, or another way to say, ways to avoid osmosis and confusion. Okay, always start with osmosis, and remember that water always moves from a higher water potential to a lower water potential. Draw it, sketch the problem, and use it to determine where there's more water, where the higher water potential is, and then draw arrows to indicate the direction of water movement. Learn and understand the meaning of hypo, iso, and hypertonic solutions. And remember, you can only use these terms when you're comparing solutions. Remember that a hypertonic solution has a lower water potential, whereas a hypotonic solution has a higher water potential. Pure water has a water potential of 0, and water potential is negative for all solutions due to the solutes, right? So as soon as you add solute, it's going to make the water potential more negative. Okay, so if you stayed tuned in for the whole lesson, you would know that the correct answer is the one scientist that I never got to meet was Dr. Franklin. And my students know that she's one of my personal heroes, and sadly she passed away before I was born. And I wish that I would have had a chance to thank her for her work on behalf of all scientists, especially on behalf of women scientists. So here's your homework. All right, so what you have here is more practice with this idea of determining the molar concentration and then calculating the water potential. So I've given you a data table and a graph. You have a temperature there, and you are asked to determine the molar concentration of the cell and then calculate the water potential. Okay, so tomorrow this will be your warm-up, and my colleague, Ms. Pfizer, will take you through that as your warm-up. One note from College Board, and that test exam date is getting closer. So if you need either mobile tools or connectivity, or you know someone who does, maybe one of your friends does, reach out directly to College Board and let us know, collegeboard.org/tech, and please do this by April 24th. Thanks for watching. I hope you join us again. I hope you join us for every lesson. Be sure to complete that practice question. It'll get reviewed in the next lesson, which is on enzymes. So have a great rest of your day, and I will see you again on Thursday.